So I have had quite a few experiences, and I have a tendency to try to put a lot of them into a talk. So what I would like to do here is to share with you a number of different things that kind of a culmination of what I have learned in my career with bees and beekeeping over the past 30 years. And the title is Managing Maladies, which, you know, is all the things like foul brood and chalk brood and sac brood and mites and beetles and, in our case, bears. You don't have to deal with deal with bears in Pennsylvania. And I would argue that maybe managing these things by letting them alone, may be a better approach than some of the things that we've been trying to do. I also call this nature versus nurses. And I would argue that our beekeeping today, a lot of us have become nurses to our bees, as opposed to the bees that are able, actually on their own, to survive and do quite nicely. So I'm going to tell you the take-home message of this talk first. And that is that as beekeepers, we really need to employ an IPM approach to managing pests and diseases in our colonies, or I would argue, do nothing at all. Do nothing at all. And this is, I gave this talk actually in Maui a couple of weeks ago, and for them, this is in Hawaii. They have it's an island, of course, and they don't have varroa, but they will have, because 2 of the islands in the island chain have varroa and several don't. They all have hive beetles, though, and so they're really lucky. They're in a position to really be able to do nothing at all and benefit from the outcome of that. In our case, I would say that an IPM approach might be better than doing nothing at all, but for some of us, doing nothing at all is working. All right. The second thing I would like to mention, or the second take-home message here, is that we all need to advocate as beekeepers, people who enjoy bees and beekeeping and want to protect our bees, and also, you know, most of us in this room, I think, would want to have a food system that is safe, not only for our bees, but also for us, an agricultural system that is safe for the bees. And it's not. It's currently not safe for the bees, and I would question whether it's safe for us. And I'm going to show you some data that's going to hopefully express that, give you the evidence for that. Integrated pest management. Okay, integrated pest management. And I'm going to talk about that more in detail. But that we all need to advocate for an integrated pest management approach in terms of strategies for controlling pests in our food system, essentially. And that's not happening right now. So that's the take-home message. And now I'm going to give you the evidence and share with you why this is so, and we'll get back to this at the end. So, but before we do that, we're going to take a little trip to Africa, to Kenya in particular. When I was in Sudan in the 80s, this was before we had varroa mite in the U.S. It was just before. I went to Sudan, and the things that we were really focused on was mainly American foul brood. That was the biggest issue that we dealt with in the early and mid-'80s until we had varroa mite. And so when I went to Sudan, I was really surprised that we never saw a brood disease while we were there. And this always intrigued me, and I always wondered about this. I also got to teach a course in Uganda, and I was only there for a couple of weeks, so it wasn't like I saw a lot of bees there, but there was not brood disease. And then in 2009, we had the opportunity to go back to Kenya and do some work there. And I'm going to talk to you about that a little bit. But again, our colleagues there at the International Center for Insect Ecology and Physiology confirmed that, yeah, brood diseases aren't an issue here. We don't have brood diseases. So, and it just was pretty amazing that there are no brood diseases there. What they also told us was, before we went, we don't have varroa mites here. There's no varroa mite in East Africa. So we went, and first time we looked in, we found varroa mite. And it turned out that varroa mite, we very quickly did a survey all around the country, and we found out that there was varroa mite in every hive. And that wasn't so surprising, maybe. What was more surprising was that people didn't realize it. It wasn't an issue. Varroa mite for them was not an issue. So here we are. This is a number of us in 2009 in Kenya. We're doing some work with hive beetles. They do have a lot of hive beetles there, and they have issues with hive beetles. That is one of the things that they do struggle with. This is one of our graduate students doing a sucrose sensitivity test. Is it possible to turn the lights down maybe just a little bit up front here?
Yep.
I don't know if you can see those slides as well as you might if the lights are a little lower. This is our very good colleague, Elliot Mooley, who's, you can see here what he's doing is he's doing the hygienic behavior test, thank you, on a frame of brood, and at the same time he's uncapping the drone brood because the reason why they didn't realize, this is kind of interesting, that they had varroa mite is they have something else that we used to have, we used to see a lot of, and that's the wingless fly, Borreliaceae. I don't know that, probably none of you have ever seen it. But it was quite common, particularly in the South in the 80s. And now that we've been treating so much for varroa, we don't see Borreliaceae. It's not really an issue for bees. It feeds on the nectar that when they do trophallaxis, when they share food, one bee to the next. This little wingless fly in the immature stage runs down on the mouth parts of the bees and feeds on that nectar that's being exchanged. And that Borreliaceae looks a lot like varroa mites, only it's an insect and not a mite, so it only has 6 legs instead of 8, right? This is doing a hygienic behavior test, or excuse me, this is doing the sucrose sensitivity test using the proboscis extension reflex. Very interesting assay. We use this sometimes with pesticides to determine if bees have any issues with memory and learning. So are you familiar with this proboscis extension reflex test? No. Okay, so what you do is you put put lots of bees here's a lot of bees, in these little jackets, it doesn't hurt the bee, and you feed them sugar syrup, and at the same time you present an odor to them. And they learn very quickly to associate the odor with the reward of the sugar syrup. And so then you can give them the odor without the sugar reward, and they'll immediately put out their tongues. So it's a way to test how quickly they learn to associate the reward and the odor, and how long they remember it. And one of our students did some work with this using this assay. It's called, again, a proboscis extension reflex assay, and we can feed bees things like pesticides, sadly, and then we can test whether or not it interferes with their memory and learning using this assay.
And does it?
Yes, it does. Even the inert ingredients that are used to added to pesticides that are supposedly not active, some of those can also interfere with memory and learning. So we have some really good evidence for that. So this was the first time varroa mite was identified in East Africa, and we found it, again, in most of the country, not all of the country. I'll show you a map of our findings, and also in part of Tanzania and in Ghana, which is actually in West Africa, where it was not found before. So again, in Africa, varroa's there, and yet it's not like it was when it was here. You know, here it has devastated, and in many places where it's found, it absolutely devastated the hives, right? We had large losses of beehives when mites were first found. We introduced chemicals to control those mites, and when the mites became resistant to those chemicals, we saw increased losses again. So this has been a big problem for us. Varroa has been, and it is
drone brood, isn't it? Yeah.
It is drone brood. That's correct. That's correct. So after this happened, we got some funding to do a very large-scale look at the effect of varroa mites on bees in Africa, on African bees. And these bees, by the way, this Apis mellifera scutellata, the same species of bee that we have, the same as the European honeybees, but it's a different subspecies. And in Africa, it's like in Europe. Here it's all big mishmash, right? Because bees aren't native here. But there are separate populations in Europe that are specifically adapted to environments in Europe. And the same is true in Africa, only even more so, because the bees haven't been moved around like they've done in Europe, and certainly not like we've done here. So in Africa, they have very, very distinct subpopulations. So this Apis mellifera scutellata is the savanna bee, and it is the parent of the bee that was taken to South America, identified as the killer bee or the Africanized bee, and that is now here, of course, in Florida as well. So these are the parents of those bees. So we went around the country and...
Are they as aggressive?
Yeah, yes, yes, they are in general. Again, all these things, it's a population, so there's a lot of variability in a population. But in general, these bees are much more defensive of their nests. And it's because over time in Africa, instead of bees being managed like they were in Europe for thousands of years, bees were hunted in Africa. So if you're, a population that's being hunted, which are the bees that are going to survive in that environment? The ones that are the most aggressive, that's right. And if you're gentle and very, you're going to succumb pretty quickly to the hunters, right? So
when you say they hunt, they destroy the bee to get to the honey?
So that's a good question. The question is, when they hunt, do they destroy the bees to get to the honey? And in many cases, yes, they do. It's very destructive. But more recently, honey hunting has become more sensitive, I would say, depending on where you are in Africa and how much knowledge people have. They'll tell you that they don't take the brood, they won't harm the brood. And these bees also are very good at absconding. So once they're disturbed, they reorganize and they abscond. They just leave. That's another characteristic that's very common. So oftentimes the colonies do survive, even though they are sometimes pretty badly, the honey is removed and even the brood. Brood is actually quite common that people would eat brood or use brood for brewing beer, that kind of thing. So it's an important protein source. Insects in Africa are an important protein source. So we went, and if you have a question, please feel free to ask me. It's fine for us to be, casual. So we went around to lots of different places and we assessed the colonies. We looked at the hives and we looked at the adults and the brood, and we looked at the stores, did varroa mite sugar shakes, tested some of them for hygienic behavior. We interviewed the beekeepers. There's Muli and myself interviewing a couple of beekeepers along the coast. So hygienic behavior is that slide I was showing you before. So I'm sorry, I sometimes am assuming you know things that, because so many of you are new, you might not know. So what we do is a really good characteristic for bees is their ability to remove dead and dying brood or sick brood, infested brood. So we actually take a section of brood and older brood, and we use this PCV pipe, and we pour in liquid nitrogen, which freezes and kills all that brood. And then we put that frame back in the hive for 24 hours, and we look at how good they are at removing that dead brood. A hygienic colony will remove 95% of that brood in 24 hours. And so that's what we're looking for, is how hygienic. Are these bees just really good hygienic bees? Is that part of why they're able to resist this varroa mite? We know that hygienic behavior also is associated with the ability to detect and remove brood infested with varroa mite. So it's a really good characteristic for bees to have. Okay, so we also did a lot of sampling for the population structure, so how many hives were colonized in a particular area. We took samples for viruses and also for other diseases as well. So this is what we found, which was interesting. So these are the populations that we looked at. The blue here is varroa mites, where we found varroa mites, and the white is where we did not find varroa mites. So this up here, this white dot, that's across a huge desert. And that was the one place where we did not find varroa mites, and that's right up against the border with Somalia. And I can tell you, we did not go there. But our Kenyan colleague, caught a plane and went up there and did the sampling for the bees up in that area. And that's a different subspecies. That's called Yementica. It's the desert bee. It's a desert subspecies. So we sampled here along the coast. We see smaller populations of varroa, a lot here in Nairobi. The highest populations of mites were in Nairobi. And that becomes interesting. In a minute, I'll tell you why. And then in the western part of the country, we found quite a bit of varroa. Here we're looking for viruses. So we sampled 5 colonies at each location. And interestingly enough, no varroa, no virus. Here, we had very little varroa, and we had, in these particular colonies, in this apiary, no virus. And of the 7 viruses we looked for, we only found 3, which is also interesting in and of itself, and very little nosema. Only 2 places where we found nosema. So again, these bees have less virus. In general, the varroa mite levels are pretty low, and there's very little nosema, which was, again, interesting to us. And no brood disease. However, some of the places we visited, they did have, this is 200 hives, and there was only 6 bee colonies that were colonized there. The colony sizes were small, and the honey production was kind of low, and people were complaining about this. So it did look like there were some things going on. From our conclusions from this project, all this sampling and the projects were that honeybees in Kenya do seem to have some issues with health and productivity, at least in this window that we looked at them. But we didn't really know why, because the overall, the mites seemed to be lower, the mite levels were lower. There were, again, fewer viruses, less nosema than in European and our bees here in the U.S. Varroa mite levels were correlated with elevation, and we've known this from other studies that the higher the elevation, the higher the mite loads. And we think this has a relationship. There's some relationship then there to environment. Environment's playing a role. These places also are, terribly fraught with droughts. Some years they have really poor bad droughts, and they have poor honey flows. And so was this something in the cycle of what was happening with bees over time? There's not much historical data here, so it's really hard to know exactly what's going on. But it did look to us like varroa was not, and mite and diseases were not playing that big of a role, and certainly they weren't killing colonies. We certainly looked at a lot of colonies that were quite healthy, quite strong. Here's one location where you can see the bees were very, very healthy and strong, lots of honey production. I was there just in January. This is the first rainy season, and they usually don't make honey until the second rainy season, which happens like in April, May, and then they harvest in June. But all of these hives, these are traditional log hives. 95% of the honey in Kenya still comes from traditional log hives. And when the bees are outside of the hive like this, that's when they know that it's time to harvest the honey. The colonies are so full that the bees are hanging outside. And so we saw this in a lot of colonies when I was there in January. And this is a really good sign. It means the bees have not only colonized these logs, but they've filled them up with honey and actually can be harvested at this time of the year.
Excuse me, Maryann. If you don't harvest the honey, will they all abscond, swarm?
Well, they'll swarm, definitely. They will swarm, absolutely. And
Leave the honey behind.
well, not if they swarm. So if they swarm, they're just going to divide. So, a number of colonies, and these colonies swarm a lot. So they reproduce a lot. Swarming, of course, is reproduction. This is a superorganism, so one bee by itself is not reproduction. Reproduction is when the colony divides and you get a new colony, right? So these colonies, in Africa, it's not uncommon for bees to swarm, to reproduce 7, 8, 9, 10, 12 times. It's what they do. It's all about reproduction for them. They're not having to store honey to overwinter. Now they do have a long dry period, so they either store honey and feed on that honey, or they abscond. They migrate just like the animals do. They migrate to higher elevations where they can have access to water and to nectar. So they have kind of 2 different strategies for getting through the dry period. Mm-hmm.
So when you go ahead and you want to go ahead and gather the honey, you're in effect having to pull out the whole hive.
So I should have showed a picture of the hive in here. So typically these bees will make their combs either this way or this way. So these beekeepers that keep bees in logs actually are pretty savvy. And they are very much more careful than the honey hunters because these are their bees, you know, so they see these hives as their bees. So they very carefully, again, their brood typically is in the middle and the honey is at either end. So they can take the lid off of this thing and they can harvest the combs until they get to the brood. And many of them would tell us, oh, we always leave 2 or 3 combs of honey for the bees. So they're very, you know, it varies, just like beekeepers here, you know, a lot of variation in how they keep their bees. But the really good beekeepers understood that if they wanted the bees to stay or if they wanted the bees to be able to reproduce, that they needed to leave the brood and they needed to leave some honey for the bees. So some of them are very, very good beekeepers even though they're keeping the bees in these logs, right? So we saw a lot of good, strong, healthy colonies. This is a very interesting project, and this came up at dinner last night. Yes, I hear you all have sponsored one of these hives. So this is Dr. Lucy King's work in Voy. I don't know if that's where your hive is. Is it in Voy? Do you know where?
We don't know yet.
You don't know where? Okay, excellent. Well, this is what one of these lines looks like. The beehives are hanging between poles, and there's actually a wire between these hives. So if this is the Elephants and Bees project. So if an elephant comes through here and tries to, this is surrounding somebody's farm. You know, they have small acreages, maybe an acre or half an acre of their produce that the elephants raid. And so if the elephants trip it or cause this wire to be vibrated, the bees will buzz and hum and make noise and maybe even come out of the hives. I sadly get to see this actually in action. I was dying to see an elephant challenge these fences. But they have a lot of good data to show that the beehive fences actually do work. And so here, what happens is they get a call from the beekeeper saying, yes, there were elephants here last night. And they'll go and they'll track, they'll look where the elephants went by their tracks and actually document, you can see here the elephants actually made an attempt to enter, but they didn't enter. So for the most part, these fences it's very exciting, and the farmers, of course, get the added benefit of the pollination from the bees and the honey as well. They make money from the honey on the bees. So this is a great project. It's very exciting. So we got to actually see this when we were there. We visited the Elephants and Bee camp in Voy.
Is that a standard Langstroth hive?
No, those are, oh, I'm sorry, this is, yes. This is one place, the place that we visited, this is a picture that Lucy gave me to use. The place where we were, they were actually using Kenyan top bar hives. Actually, they were not, I take it back, they were not using Kenyan top bar hives, they were using all Langstroth hives. Kenyan top bar hives end up not to be very good in Africa. They're not working very well in Africa. So they were all Langstroth hives, that's correct.
They don't work very well here.
Yeah.
We have given Lucy an invitation to come and speak.
Oh, that would,
So, because she's in Orlando.
uh-huh.
So would you put in a good word for
She's
her?
in Orlando now?
No,
Oh.
she
She
travels
goes
to
to
Orlando.
Oh,
Because Disney is one of her big sponsors.
Right, that's right. And when I was, if you don't get her, it may be hard to get her, but she had a intern, Matthew, who works at Disney, who's from Florida. You should get him.
Matthew, who?
I'll find out for you. I'll find out his last name. He was a very good guy. He works a lot with Disney. Disney is sponsoring him to be there. And so I got to meet him, and it was very interesting though. They are helping her, but they know nothing about beekeeping. So it's amazing what they have done without knowing anything about beekeeping. So my Kenyan colleagues, went out with them. We all went out to harvest honey. And the first hive, he saw what they did, and he said, I want to know how to do what you just did. Because they were spending the whole night. They do the harvesting at night, first of all, which you don't have to do. And they got stung a lot. You should see the way they covered themselves up. And it took them till the middle of the night. We were done in 2 hours harvesting 20 colonies, So he was amazed. Oh my God, I need to know how to do this. So he wants to learn about bees and beekeeping more. And so it would be great for you to have him here and teach him some beekeeping. All right, I will. So this is their honey, highly prized honey. They get a lot of money for this honey, and we couldn't even buy it. It was very good. We couldn't even buy it. They had it all sold before they even. So the beekeepers are making out really well. Okay, so our question is then: are African bee populations better able to tolerate pests and diseases, and if so, why? So in Africa, the populations of bees are very old. They're native to Africa. They're essentially wild colonies. What people do is put out nesting cavities for them, these boxes. And the bees come, they make some honey, the beekeepers harvest, the bees typically go. Or they do stay over the dry season if they have the resources that they need. They have a population structure, so their bees are adapted to the areas where they are. Think about us in the U.S., in New Zealand, Australia. Our bee populations are new. They're not old. We've imported bees. They've just been here for a couple hundred years. They are not native. They are highly managed, and we have little population structure. So it's a very different scenario. And I would argue also, in general, now this is very much in general, that our beekeeping in this country is economically driven. We want honey, we want pollination, it's all about getting as much as we can from these bees. And I'm not speaking about, a lot of you in this room are, backyard beekeepers. I don't think there's any commercial beekeepers here.
Yeah, there is.
Well, okay, even if there are, in general, I would say in general in this country, we have an economically driven system as opposed to what happens in Kenya and in most of Africa. It's biologically driven.
Unfortunately, the whole country is economically.
Well, that's exactly true. That's exactly true. It is a way of life for us in this country, and I'm not saying it's wrong, but it has its advantages and its disadvantages, right?
Well put.
Okay, so biologically driven is interesting because the bees now are in charge of their own fate, right? Biology is driving the system. And so in our system, it's driven by humans, by us. Again, honey production, putting bees on trucks for pollination. We need bees in our agricultural systems for pollination, which is the way our system has evolved. So this is all about the bees having to adapt to the way that we keep them, right? And we keep them in apiaries. This is not how they normally exist. We keep them close to each other in an apiary because it makes it easier for us. We, again, truck them around for pollination. When they're sick, we feed them antibiotics or treatments for mites. We try to keep them from swarming, right? Because if we keep them from swarming, they make more honey for us. Yet it's their natural behavior to swarm, and swarming may do things for them other than even increase their population that we don't necessarily count on. So all the things that we're doing are really to make life easier for us. And in some regard, this has been successful, right? If you look at honey production in this country, we have a very high yield per colony, and it's been relatively high over a long period of time, between 50 to 80 pounds per hive. That's a lot of honey. They don't get this much honey from their hives in Africa, I can tell you that. But I would argue that this has come at a cost, right? It has come at a cost. And so if we look at, in 2007, the American Academies of Science put out this publication, The Status of North American Pollinators. Not only honeybees, but bees in general are in decline. And we all know this. We all know that this is the case. We have issues with the health of our bees. There are lots of other evidence since then to support the fact that not only honeybees, but bumblebees and solitary bees, even butterflies and some beetle species that are pollinators are in trouble. So again, this idea that when we transition from wild to apiary, it has a number of different disadvantages that I already described to you, right? Including increased exposure to pesticides. We use the same combs over and over again, so any pathogens that are in there, any pesticides that are in there accumulate in our hives, as well as the other things that I mentioned. Of course, we use bees for pollination. Monocultures have come about in part because we can take bees for pollination, and they're available for this, and so now these things are dependent on one another. It's like a chicken-egg kind of argument.
Could you, you lost me a little bit there. What do you mean exactly? Could you define monocultures?
So a monoculture, is a very large acreage of one crop. So here we have in Adams County apples, right? Apples, apples, apples as far as you can see. Here we have, this is also in Pennsylvania, this is hundreds of acres of pumpkins. And these are colonies that are being ready to be moved into almonds in California. So these are thousands of colonies. And when these bees are in almonds in California, two things happen. It's a monoculture, so they have no access to anything other than almond pollen and nectar, right? So not only is there issue with potentially with pesticide exposure in a monoculture, there's also no diversity of food. And one of the things we absolutely know about honeybees, have any of you ever used a pollen trap or looked at a frame of pollen? What's it look like? Is it one solid color? That's because bees must have diverse sources of pollen to be healthy. They must have diverse sources of pollen to be healthy. Not all the amino acids that they need are present in any one kind of pollen. So they have to have diverse sources of pollen, and they will go to great lengths to get it. In Adams County, we put hives in Adams County for apple pollination for a pesticide experiment we were doing. When we look at the pollen and we analyze the pollen, even though they are surrounded by apple blossoms, which produce both nectar and pollen, the pollen in those samples varies from 16 to like 55% apple pollen. Right? They are working to find other sources of pollen because they need it. So that's what they're going to do. That's what they're going to do. And this works to our advantage as well as to our disadvantage. And we're trying to protect them. So I want to tell you a little bit about some work that we did whenever the whole CCD thing occurred. You all know what CCD is, right? Colony collapse disorder, right? So when that happened in 2000 and at the end of 2006, the beekeeper that sort of brought that to the attention of the public was Dave Hackenberg. And he's a Pennsylvania and Florida beekeeper. And he came to us in a state of panic and told us what had happened. So of course, a bunch of us mobilized, and we got money from the National Honey Board, and we did a lot of different things to try to understand what was causing CCD and even pollinator decline. And it so happened that there was a toxicologist in our department named Chris Mullen, who is quite good at toxicology and has worked with pesticides all his career. And he came to me and he said, do you think this might be a pesticide problem and maybe even a neonicotinoid problem? I've been reading what's going on in France. I said, yeah, we should look at this. We should absolutely look at this. So he and I teamed up, and later Jim Frazier, my husband, joined us as a team. And we did an analysis of thousands of samples. Well, not thousands. We did 1,200 samples. You'll see the data from 1,200 samples. And we worked with Gastonia, North Carolina, a lab that can actually analyze for pesticides. We have a screen for 171 pesticides, and they can look at the part per billion level. And so in these early samples, this was from 2007 to 2012, this is 1,200 samples that we looked at. And this is the list of pesticides that we found in those samples. Okay? So this was pretty shocking to us to see this list of pesticides. And there are pesticides in here, not just neonicotinoids, which everybody is like really focused on, not just imidacloprid, but even pesticides that are no longer legal to use in this country anymore, that have been illegal for a long period of time, right? it turns out bees are really good at scouring their environment and bringing back into the hive things that are in the environment and are probably recycling in the environment, like DDT.
That one doesn't break down.
Right, right. It just keeps getting recycled. So this is pretty sobering and pretty, I can hear the gasp from all of you. But it's important to consider that while this is a lot of different pesticides, how much are we actually talking about? Is this like 2 parts per billion one time that we found it? You know, so how often do we find it and at what levels and how toxic is it to bees are all questions that we need to ask around this list, right? So what was even more troublesome to us was this. So 137 pesticides were detected, so it's a great diversity of pesticides, and it shows how good bees are at picking up things in their environment. Only 7% had no detections. On average, in a pollen sample, we found 5.4 pesticides per pollen sample. We found one pollen sample that had 31 different pesticides in the pollen sample and 39 in a single wax sample. Question?
I don't understand the first 2 bullet points, the
So this list right here, okay? So out of the 1,200 samples we looked at, this is 139 pesticides. That's just the total number that we found, okay? And of the 1,200, only 7% had no pesticides at all. Does that make sense? So only a very small amount of samples that we collected had no pesticide. So what it's saying is that in general, our bees are being exposed to pesticides. Now,
Is that strictly pollen that you were testing?
we looked at pollen and wax, okay? We also looked at bees, adult bees. We looked at flowers. We looked at foundation, old foundation, as well as new foundation, pre-varroa mite. The thing about this is that most of these pesticides are lipid soluble. So they go into lipids. They want to dissolve in a fat. So that's why we find them in wax and in pollen. The USDA lab that was doing this work for us kind of panicked when they saw this. And they immediately started looking at honey. And it turns out that honey has very little pesticide because it's water-based. So the pesticides are basically being sucked out into the pollen and into the wax, which is, on one hand, a good thing. If you're making lip balm from wax, it may not be a good thing. Or if you're selling comb honey, it may not be such a good thing.
Or if you're feeding.
What part of the country were the samples collected?
What part? All over the country, all over the U.S. Yeah. So we had a lot of migratory bee samples. We also had a pesticide cost sharing program where any beekeeper could send us a sample and we would pay half the cost. And it was funded by the APIS-M, Project APIS-M. They put the money toward it. And so a lot of beekeepers from all over, small beekeepers who were making foundation, or not so much foundation, but products from their wax, who were selling comb honey were concerned. So this represents most, I would say, I should probably have a figure on this, how many were commercial beekeepers and how many were not. Most of them, I would say, were commercial beekeepers. But it does represent beekeepers from all over
With
the country.
7%.
7%?
Ones that had no detections, were those in concentrated areas?
No, not at all. No.
Now, the comb honey, that's usually a fresh honey. It's not been used or recycled to find
Yeah, well,
much in there compared to
again,
a
this
brood
stuff.
comb that's 3-4 years old.
So that he's asking about the comb honey, which has newly secreted wax. So a beekeeper sent me his wax cappings, and he had only used Coumaphos and fluvalinate a few times, in the past. I mean, I trusted what he said. He's somebody that I know quite well.
Right.
And it was amazing how much Coumaphos we found in the cappings. So this wax is and the pesticide is being circulated through the colony.
So
And
it's
when
absorbed, what you said sucked, what do you?
it's absorbed into the wax,
Yeah.
right.
But I mean,
And-
comb honey is made and then chopped out and got.
Right,
Yeah. so
right.
It's different than somebody that's been using the Coumaphos at the same time that the bees are making the honey.
So
or-
no, he was using Coumaphos like years ago in his brood nests.
Okay.
And this was capping's wax from several years later, when he didn't use the coumaphos and fluvalinate for several years, and we still found it in his wax. The problem is, and I can tell you, if you buy foundation, wax foundation, it has coumaphos and fluvalinate in it. We looked at all the coumaphos, and I should be careful because we did this about 5 years ago, and we haven't done it recently. But every bit of wax that we, from every supplier, had coumaphos and fluvalinate in it. Those are the materials that are typically used for varroa mite control. So,
But if you're a treatment-free beekeeper, you don't use that, and you make your own wax foundation, or you use your own wax, typically it's not gonna be there.
if you do make your own foundation, that's true. But how many people make their own foundation?
Most of it's plastic.
So this is
Or
what
we
we
let
do.
bees make it.
Yeah, we use plastic foundation without wax, and we coat it with our own wax. That's the easiest thing to do if you want to not have a foundation of
So the
pesticides.
plastic-free wax has probably got.
Absolutely, it does. We looked at that. We looked at
Oh,
that.
wow.
It has
Okay.
pesticides in it too.
You're talking about the stuff that's right there. That's the best
Yep.
stuff.
Probably.
Yep.
Probably. I can't say for sure. Again, I should be careful and we should relook at it. But when we looked at it 5 years ago, everything had coumaphos and fluvalinate.
When you say your own wax, is it from your own bees?
Right, from our own bees. Or from Africa. We got a lot of wax in Africa and it had no coumaphos and fluvalinate. And no,
They don't have the money to use it.
exactly right. Exactly right. Exactly
They
right.
don't have the problem to use it.
So, a question back here.
Did you say that the plastic foundation also has pesticides?
So the plastic foundation that has wax coating on it.
Wax coated.
Right, that's wax coated. Yeah, yeah. So,
Aren't those all wax coated?
I don't know what those are. I have no idea.
Chances are.
I have no idea.
How come it's not transfer over to the honey itself?
Because honey is liquid, is water-based, and these pesticides, for the most part, are lipid soluble. They want to dissolve in a fat. That's why DDT, where they found it was in mother's milk, in mammary glands, because they're fat. It's a fat content, and so this stuff is dissolving in fat. It doesn't want to dissolve in a liquid, in water. That makes sense to everybody?
So hypothetically, if you were to buy a pre-treated, or you don't know if it's been treated with a wax, can you use your own treatment-free wax and obliterate that?
So you can take your foundation, plastic foundation, without any wax on it.
Well, what if we don't know that,
Yeah.
and we used our own wax to wipe it out?
Most all the foundation you buy is pre-waxed, most of it.
Right.
But,
And
so
that's the
because we
specific
don't know, if we take our own wax as a precaution and add another layer on it,
It's not gonna do anything.
Yeah.
Just walk it.
It's amazing how much they recycle. The bees themselves recycle wax in the hive. So if you recoat it, what I would suggest is that you scrape it off, scrape your wax. We do this every year. We scrape.
Put it in a wax moth farm.
Yeah, that
That's
helps.
wet.
That helps. And then you can recoat it, but basically you probably have a little bit in there, but
You could
all
drive
this
yourself
stuff.
nuts.
You decide how much risk you're willing to take.
I've started with mine, I had to start with foundation, because that's the only way I could get bees. But I'm using, in my hives, I'm using just straight top bar.
That's another thing people do is they just use either
Goes a lot
stretch.
slower.
Yep. Or
Goes a lot
no.
slower,
Yep.
but-
No foundation at all. Yep. That's right. That's what a lot of people are doing. Okay, I'm going to try to get through these couple of
Yeah,
slides,
go ahead.
and then I'm going to let you ask me as many questions as you want. Okay. This is a study that we did to look at in 8 different crops, because we knew that a lot of these pesticides, other than the miticides, were coming from crop pollination. So this is 8 different crops that we looked at, and we took samples of the flowers, the trapped pollen, the bees that were returning that were live, and then the dead and dying foragers around the hives. And so what you can see, just for instance, cotton there, we had in the cotton flower 4 pesticides, none of which were toxic to bees. But when you look over here at the dead and dying bees around those hives, they had 11 pesticides, 6 of which are toxic to bees. And so if we look at a cotton or a corn tassel near that crop, or within that crop, like a volunteer, there were 21 pesticides, 8 of which are toxic to bees. So what this in part is telling us is that, first of all, bees are being exposed to a lot of pesticides, particularly in cotton, alfalfa, cotton, alfalfa, pumpkins, and in almonds. But not all the pesticides are coming from the crop itself. Again, it indicates that these bees are going beyond the crop and picking up pollen from other places that's contaminated. So even if the grower is working with the beekeeper on the bee-pollinated crop is trying to be careful, bees, do you know how many acres the average beehive covers? What's the average foraging range of a beehive? How many acres is that? 3 acres is 28,000, 3 miles is 28,000 acres. We know that if bees are stressed, they can go as far as 10 miles. That's 77,000 acres. How do you know what's happening in 28,000 acre range within your hive, within your apiary?
You don't.
Yeah, that's right.
That's why when I hear organic honey, I laugh.
Yeah. So
Best you can, that's all you can do.
So pesticides include insecticides, herbicides, fungicides, and again, we found all of these in these samples. We found all the classes of insecticides, even again those that are no longer legal to use, like the chlorinated chlorinated cyclodienes and the organochlorines. And we found herbicides and fungicides. We found metabolites and inert ingredients. And all of these things are problematic for bees. Fungicides, we now know bees have a lot of microbes in their our good biota, good microbial community in their guts and also in the transformation of pollen to bee bread. And these fungicides are interfering or killing those good microbes. The thing about fungicides is they're not toxic to adult bees, so growers apply them during bloom. So this is what we found the highest levels. While we find coumaphos and fluvalinate most often detected in wax and pollen, it was fungicides that we found at the highest levels because there's so much fungicide application during bloom when bees are pollinating crops. Just go through that. Again, herbicides, the problem with herbicide, there is so much herbicide being used in this country, it's unbelievable. And herbicides are reducing the variety of plants that are available for bees to the forage that's necessary. Bees are, again, polylectic. They need a diverse source of pollen. And this is what pollen looks like we had a bunch of pollen trapped from a colony and we were feeding it back, and you can just see the variety of plants that they were collecting the pollen from based on the colors in that pollen. And so again, I already told you this, but even in apples, where there's all that apple bloom, it's highly variable how much apple pollen is in these colonies, and the bees are going after other kinds of pollen. But the growers are, getting rid of the weeds and the other kinds of plants used with the use of herbicides. And there's the USGS, I don't think I have it in here, the USGS, US Geological Survey, is a great site to go to show you how much pesticide over time is being used by the pesticide. And if you want a real shock, look at glyphosate. It's unbelievable how much glyphosate has increased. It has, I think, doubled. Usage has doubled since 2003. Okay, so pesticides alone, of course, are not responsible for the losses that we are experiencing. It's a very complex picture. We call this the 4 Ps. You've probably heard of this. Parasites, meaning particularly varroa mites, but also tracheomites, pathogens, poor nutrition, and pesticides are all involved in pollinator decline, particularly impacting our honeybees. And the thing about this is we know that these things interact with each other. So pathogens, we know, can actually result in poor nutrition, pesticides can result in poor nutrition because we're eliminating so many of our diversity of plants with herbicides. The parasites can actually carry vector pathogens. We know that poor nutrition can actually immune suppress bees that then allow pathogens to be deadly to the colonies. And we know that pesticides can also suppress immune function and cause pathogens to be a problem. And lastly, we know that these pesticides are accumulating in wax and are present over time and build up in colonies, causing problems. Okay, so back to Africa, again, this biologically based system where the beekeepers the humans are adapting. I want to share, I mean, so for me, a big part of this pesticide thing, maybe not so much for you as a group, because I think a lot of you are how many of you are trying to be pesticide free, not use coumaphos or fluvalinate? Okay, so a lot of you. Treatment free. Okay, so that's great. But a lot of people are not treatment free, and a lot of people are continuing to try to use pesticides to control their mites. And I would argue that in the north, you know, we tried for years not to use anything to control our mites because we were doing pesticide work. And every year we had like 5 or 10% survival. We started using not oxalic acid, but formic acid, and now we have 70 to 80% survival. So, you know, I had a really interesting friend of mine who said to me, she's trying very hard in the north to be pesticide free too, treatment free. And she said to me, you know, though, if my dog has fleas, am I going to let my dog suffer from fleas? No, I'm not going to let my dog suffer from fleas. Why should I let my bees suffer from mites? So, eat the dog? I would argue that that's why I argue for an integrated pest management approach to mite control. All right. So I just want to point out this one last thing about Africa before we leave Africa. And this is what happened in South Africa. In South Africa, this is a scientist named Michael Alsop, and in 2003, he was saying 2 problems in South Africa, the capensis problem, which is a really interesting problem in and of itself, which are a special subspecies of honeybee that can actually, the females can produce female eggs. Not fertilized, but female eggs. And it wreaks havoc with the bees that can't do that. So I'm not going to go into that, but that problem, along with varroa mite, have severely curtailed beekeeping in the country and may well have an impact on bees and beekeeping throughout the continent. But by 2006, that was published in 2003, and by 2006, he's saying in both wild and managed populations of honeybees, varroa appears to have been reduced to the status of incidental pest. Yeah, how? That's exactly right. And he says it's because they've developed resistance to the mites, and it's because the bees are very hygienic and because we have not used chemical controls. All right? So when we look at populations of bees in Kenya, this is really interesting. So this is the hygienic test. Again, I just described it to you. And here you can see a colony that's not very hygienic, and here you see one that is very hygienic. It's removed all of that dead brood. 2 different locations where we did this on 10 colonies, and we saw very different results. So Isipi, which is the International Center for Insect Physiology and Ecology, which is in Nairobi, and then Mwengi, which is an area where there's very, very not very much managed beekeeping. It's these wild colonies that are kept by beekeepers, but it's a really good honey producing area. And based on some genetic studies that one of our partners did, he characterized these populations of honeybees, because we were trying to look at, is there a difference between the Monticola and the Apis mellifera scutellata and the Latoria? And what he found was there wasn't much difference in their genetic makeup, except this one population here, that they all kind of grouped together, this population here. And it turns out this is the Nairobi population. And what we think has happened is that Europeans, whenever they went to, the English, the British, and the Europeans, when they went to Kenya, they went to Nairobi, a lot of them. And they brought bees there with them. And we think that these are European bees, and these are the bees that seem to have the highest level of varroa mites and the biggest, the most viruses. So that is not the native African bees. The native African bees seem to be the most hygienic and the most resistant to mites and diseases. There's lots of reasons why we think the bees do seem to have resistance to diseases and mites. One is that they have this migratory swarming, that they reproduce a lot, that they abscond a lot. But we also think that they have physiological mechanisms like, again, hygienic behavior, and which is also a behavior actually, but that allows them to be resistant to mites and diseases. So what we would really like to do is utilize our knowledge of these populations, our understanding of these wild populations and how they are resisting the mites. And as a matter of fact, you all might be taking advantage of that very thing already. You have African genes in your population here. And it may be part of the reason why you're able to be treatment-free here is because you have African genes in your population. So it may be a really, really good thing. And we'd like to also help each other. We would like to help them with their bee management and help them do a little better job with some aspects of bees and beekeeping in Kenya. But can we also learn from them and help ourselves? So what makes sense for the bees and the humans? We want this to be sustainable for bees and humans. And again, this is why I advocate for an IPM approach. If you don't need to treat, as it seems that you don't treat. If we need to treat in the north, okay, treat, but treat when we need to treat, not prophylactically, and treat with something that is not going to build up in our wax and be problematic to the bees in the long run. We have a number of different tools now. We have screen bottom boards, we have some resistant stock, we have formic acid, oxalic acid. We don't have to use things like coumaphos, fluvalinate, and other kinds of materials, amitrez.
Those two acids, they don't build up in the wax? Or
As far as we know, they do not.
the formic acid's already in the hive, from what I
Right,
understand.
that's right. What we see now is a lot of thymol. More recently, we see thymol that does seem to build up in the wax.
Right.
But the acids we do not see. We do not see. Again, integrated pest management, I just want to quickly go over this. It's the objective is no longer to eliminate the pest, and I'm talking in general here, whether we're talking about agricultural systems, gardens, fleas on dogs, although nobody wants fleas on their dogs, even if it's just 2 or 3, right? The objective here is to manage the pest by keeping it below an economically damaging level. That's what we're trying to do. So I would argue that you use integrated pest management every day, every single one of you in this room. Is there anybody here who does not have screens on their doors or on their windows? Anybody? Right? A screen is an integrated pest management approach to fly control. We could spray pesticides on the flies that come into our house. We could spray Raid, but we don't, right? We exclude them. And that's what integrated pest management does. It tries to take those kinds of steps to minimize the pests that we face. Okay, so the key elements here are that it's knowledge intensive. We do have to understand the behavior of the pest and the host. It integrates a variety of tactics. There's a toolbox that we use, not just one treatment year after year after year. It's based on thresholds, so we have to know at what level these mites are going to kill my colony or cause my colony not to be productive. We take into consideration environmental factors and economic factors. You can't use a tactic you could if you're not thinking about making money from your bees. If you just like your bees and want them not to die, then maybe it doesn't matter how much money you spend on your bees, right? In general, IPM also requires educated consumer. If you go to the grocery store and you pick out 12 perfect oranges and you ignore the oranges that have a little bit of damage to them, insect damage or pathogen damage, what are you saying to the grower?
Go to it.
Please put pesticides on my food because I want the perfect looking fruit. I'm not going to buy the fruit that has a little bit of damage on it, right? That's what we're saying to the growers by doing that. So this is why growers use pesticides, because that's what's selling. That's what the consumer wants, or that's what we're saying we want. So we all have to be sensitive about this, and we need to vote with our pocketbooks, right? We need to vote with our pocketbooks. We need to accept some damage, some insect damage to our fruits and vegetables and be willing to buy that. That's why people buy organic or sustainably grown food, right? So this idea of educated consumers is really important. And we as beekeepers need to share this information. Obviously, you all are sensitive to this, and now it has a big impact on our bees, and so it's really important for us to get this message across to the consumer.
Maryann, does that include wax? You know how a lot of fruit is waxed over or gassed? The seeds from that, can they absorb any of that?
I don't know. I don't know if the seeds can absorb the gases, or I wouldn't think the gases. No, I wouldn't think so. I wouldn't think so.
And cucumbers are dipped in wax just to keep them from deteriorating. too
Drying
fast.
out, and it's probably, you know, I mean, it depends on if you want paraffin on your. I mean, it's not like beeswax, which would be great, right? It's paraffin. It's a.
I don't know, our beeswax ain't electric.
Well, that's true. Good point. Good point. Let me just finish up here real quickly because I know it's getting late. The tactics here are highly variable. So in agricultural systems, you have things like rotation, sanitation, the time that you plant, the time that you cultivate, resistant stock. People never used to grow basil because there was a wilt on basil that you could not hardly grow basil. Now we have great resistant varieties of basil. You never see wilt. So that's a great, you know, there's a lot of good stories about resistant varieties. You have physical and mechanical, like, again, screens, like trapping, barriers, biological systems, parasites, predators. Nematodes are now being used for control of hive beetles. There's an experimental nematode. And chemicals. And the point here is that chemicals are used, they're allowed in an IPM approach, but they're the last choice. You use all of these other tactics and use chemicals. If you used IPM and for me in August, I test my bees, I do a sugar shake. If I have a threshold I have a tolerance of 5 mites per 300 bees, and I'm at 6 or 10 or 20, okay. If I'm at 6, I'm probably going to accept 6. But if I have 20, I think, my gosh, I got to do something for these bees or they're not going to survive the winter. And so I'll choose not a fluvalinate, Coumaphos, or amitraz. I'll choose a formic acid or an oxalic acid. Now we have oxalic acid. So the goal here is to use as many of these tactics as we can to keep the mite levels low, and then use these other types of interventions when we are above threshold, to monitor our threshold, and when we get above threshold, to actually use these other materials. So this is a kind of approach that I think we need to use for varroa mite control, and even for hive beetles. So I'd like to leave you with 2 things. One is this. I'd like you to read this. I'll read it quickly for you. The agricultural journals have from year to year presented through their columns various treatments as preventative of the attack or destructive to the life of the curculio, the apple moth, the squash bug, all of which, you know, are problems that we have in vegetable production in particular. The proposed treatments, we are well satisfied in the majority of instances, are as useless in application as they are ridiculous in composition. And if the work of destroying insects is to be accomplished satisfactorily, we feel confident that it will have to be the result of no chemical preparation, but of simple means directed by a knowledge of the history and habits of the pest. When do you think that was written?
About 1912.
Before chemicals?
1856. 1856. Yeah, we're slow learners. That's exactly right. And lastly, you know, we have not seen the last of our pests. Even if we figure out how to control varroa and hive beetles, there will be other pests.
June bugs.
There's Uvarroa, there's Trophalelaps, and there's this. This is the large hive beetle.
Yeah.
We call them June
It's
bugs.
like a cinch bug.
These are much bigger than June bugs. These are quite large. These are amazingly large. Here's a little better picture of them next to the bees. These are very, very large beetles.
They look like a bee.
And what, they cleaning out the brood?
Yes, Yes, this is the thing that the beekeepers have the most problem with in Kenya, are these beetles. They will live with the small hive beetles, but this is the biggest problem.
So they're gonna have to put a queen includer on the front of the door, so the beetles can't get in there.
And that's what they do, but so many of them have logs, and these logs are very inconsistent in their, so the beetles find their way in, right? Okay, I'm going to end there. That's my last slide, and I'm happy to answer questions if you have them. Thank you. Thank you very much.